Welcome to the Genetic Symposium. Today, Dr. Jorge Manni, who is co-chairing with this session with me. I am Marta Alarcón Riquelme. And we're starting. And so it's my pleasure to introduce Dr. Robert Kimberly. Dr. Kimberly is a native of New Haven, Connecticut. He received his Doctor of Medicine degree from Harvard Medical School after receiving a baccalaureate degree magna cum laude from Princeton University and a master's degree from the University of Oxford in England. Dr. Kimberly is the Howard L. Hawley Professor of Medicine at the University of Alabama in Birmingham in the United States. Dr. Kimberly is also director of the UAB Center for Clinical and Translational Science and the UAB Comprehensive Arthritis, Musculoskeletal, and Autoimmunity Center. He is a clinical rheumatologist, immunologist, and a colleague human geneticist. Dr. Kimberly?
Well, thank you very much for that very kind introduction, Marta. And thank you to all of the organizers of this meeting for having not only invited me to give a chance to introduce a number of different talks on genetics, but for organizing what I think is really a very successful meeting here in Buenos Aires. What I'd like to do is spend a few minutes talking about what the genetics of SLE and what we've learned about the genetics of SLE actually mean for patients and physicians. Here are my disclosures, and here's the outline of what I'd like to cover this afternoon, really to address 6 major points. Perhaps the first is, what are the current techniques and methodologic approaches to genetics that we're currently doing? I'd like to do this perhaps in a slightly different way than is conventionally done, and think about the different components that are needed to address the architecture of autoimmune diseases. And you can see from this slide, of course, we need a collection. We need to understand what kinds of variants we're going after. We need some sort of analysis. Through that analysis, we identify candidates, be they regions, genes, or perhaps single nucleotide polymorphisms or SNPs. And then, of course, we'd like to tie that to biology so that we can understand the pathophysiology of disease. One way to think about this is really the genetics of lupus in 3 phases. The first I'd like to call the era of MHC emphasis, where the variants were the MHC alleles. And that was tied with biology, where biology typically was wrapped in the frame of antigen presentation, although it's clearly acknowledged that there's a great deal of LD in that region, and there are probably many other candidate genes there as well. The second era was one of family-based strategies. Here the collection was a little bit different. It was multiplex families. The variants were typically microsatellites. Again, analysis which led to regions. But when one tried to tie those regions to specific biology, it was a little bit difficult. And that was in part because of the granularity of those studies. I'm sure you all know that for many years now we've been in the era of doing genome-wide SNPs with increasingly powerful technology, with increasing density of markers. And that's led to the collection of simplex families, doing case control studies as the typical design, looking for variants, which has typically been single nucleotide polymorphisms. But the analysis has been rather complex because the more features there have been on each of the chips, the greater the likelihood, the false discovery rate. And so as we transition from those statistical results to understanding real candidates in terms of SNPs and genes and regions, we've had to bring in a priori knowledge about the biology of the systems that we're dealing with. And actually, it's led to a very productive loop of analysis leading to candidates reaching over into biology, the biology coming back to further elucidate not only candidate gene regions, but specific SNPs, in order that we can understand the underlying genetic architecture of systemic lupus. And of course, this has involved both clinical rheumatologists and immunologists who are key in terms of facilitating those collections, our colleagues in statistical genetics, and those also in more basic mechanistic science, both clinical and basic immunologists. For those of you who haven't seen these machines, if this is meaningful, on your left is an instrument which allows you to scan the microarrays that are used to do the genotyping itself. This has been rather a remarkable journey. Over the last 10 years, when we've been involved through several different consortium, we've gone from chips that have in the range of 300,000 features per chip up to now over 2.5 million, and indeed I'm sure many of you know that some of the chips currently under development have more than 5 million features. So based on all of this, what are the natural variants that are considered to be risk alleles? There are a large number of genetic associations for autoimmunity which have been identified, and generally, by the nature of the design of the chips, these involve common variants of modest effect. It's also become clear that genetic risk variants often predispose to multiple autoimmune diseases. So in lupus, what does this look like? Well, the list now formally in terms of confirmed genes that are contributing to SLE risk probably exceed 40, and there are only about 20 on this particular slide, some of which you'll recognize: HLA and complement components, receptors for antibody, and a number of different signaling molecules within the cell. So what's one of the ways, perhaps a different way than simply a list in a compendium? What are some of the other ways to think about the various genetic risk variants that have been identified in SLE? Well, all of these involve a certain level of statistical analysis, and for those of you who don't recognize this gentleman, this is R.A. Fisher, who designed the Fisher exact test. And so he would think about how to put this information together, and one way to do it would be to think not only of the gene and its location and its p-value, which is in part a function of the size of the population that you're looking at, but to look at the odds ratio. And this summary, taken from a review by Andreas Sestak in Annals of Rheumatic Diseases about 2 years ago, shows you that there's a range of odds ratios from TREX1, which, although rare, has an extremely high odds ratio, perhaps as high as 25, through the various component deficiencies, where the odds ratios are between 5 and 10. And then, as you can see on the right-hand part of the slide, the odds ratios perhaps decreasing as I've listed various candidates that have been identified through the GWAS studies. One of the other principles that's been identified through these studies is that many of the associations that have been seen are in non-coding regions, and they appear to be able to regulate the quantitative effects of gene expression. This is actually very important, not only because it may reflect not only the ability perhaps of a promoter to quantitatively drive gene expression, but it also opened up another possibility, and that is epigenetic effects, which may interact with the genetic effects. Thus, a large fraction of the common variants act through quantitative changes in the thresholds for immune signaling and activation. This is a graph drawn from a review article written by Peter Gregerson and colleagues a couple of years ago, and it just demonstrates the shift in the signaling threshold back and forth according to the SNPs that are involved. And typically these SNPs may alter the fine structure of transcription factor binding sites. So of these various naturally occurring variants, what might be their relationship actually to the phenotype, or perhaps the subphenotype, sometimes called the endophenotype of SLE. You know that there are many who think that the way autoantibodies come to be is that there's abnormal handling of apoptotic material in germinal centers, and this then presents apoptotic material, or the debris from that allows selection of autoreactive B cells. So it wouldn't be a surprise then if we ask about the relationship between certain autoantibodies and variants in the genetic apparatus driving the immune system and the risk variants related to SLE, if there's an overlap between those. So in a review from Betty Sau and her colleagues, published in Annals of Rheumatic Disease this year, she's listed SLE disease phenotypes and associated candidate genes. And as we look at the top 3 rows, you can see there are a number of different variants which have at least the statistical association with the autoantibodies mentioned. But the other thing that's apparent from this slide is that there's no tight relationship between any given single variant and a particular clinical manifestations. There are statistically significant associations. A number of different variants are involved, but we're still at least a step or two removed from being able to draw the tight link between a particular variant in our genome and a particular disease manifestation. So what about the 4th question? Where are these insights taking us? I think here we've probably got the most insight that we've gotten from genome-wide association studies to date, and that is that these data can be used to infer shared and distinct pathogenetic pathways in the lupus process. So what I'd like to do in the next several slides is to borrow illustrations from a number of different reviews that have appeared in the literature, and at least begin to show you how they can aggregate into clusters of variants and gene targets which seem to make sense in terms of the pathophysiologic process. Let's start first with the TLR and the type 1 interferon pathway, and that's the one that's marked here. In red, and I'm going to go back to where I was, in that red box. So if those are some of the variants that are related to TLR biology, another way to look at it is to pull up KEGG pathways and see whether some of the constituents of the KEGG pathways have been defined as having naturally occurrent variants with statistical association with SLE. And here, illustrated by the red circles, are IRAK1, TRAF6, IRF5, and as you look through this graph, you can see a number of other variants, suggesting that the machinery of the TLR pathway, the type 1 interferon pathway, really is very important in SLE because variants in those pathways are strongly associated with SLE. Somewhat simpler diagram for that, drawn from Guerrera's review in Arthritis Research and Therapy last year, shows the NF-kappa B pathway linking TLR through A20 and I-kappa B alpha through NF-kappa B driving gene transcription. And variants in IRAK, TRAF6, and A20 are prominent in terms of those risk alleles that are associated with SLE. How about those that might be related to immune complex processing? Well, here you wouldn't be surprised to find both complement components, receptors for complement, and then receptors for antibody as well. In one illustration, there's macrophages the participation of antigen-presenting cells, T cells, B cells, then leading to plasma cells, the production of autoantibody and antigen, autoantibody complexes, which then can interact with complement and may lead either to tissue injury or further stimulation of other cells within the immune system, in this particular illustration, plasmacytoid dendritic cells. And finally, what about immune signal transduction? And here it wouldn't be a surprise, as anticipated by the graph, the conceptual graph I showed you from Peter Gregerson's review, that the threshold for signaling in both T cells and B cells appears to be altered by variants that are related to SLE. So one way to put it together is to say, okay, let's look at pathway-associated SLE candidate genes, look at the pathways illustrated on the left for you, and the genes that might be related to those pathways on the right of the slide that's being projected for you. And you can see pathways can include DNA degradation, apoptosis, and clearance of cellular debris, TLR, and type 1 interferons that we've already talked about, NF-kappa B signaling, immune complex processing, the function and signaling of both B and T cells, and neutrophil and monocyte function and signaling as well. Perhaps there's another way to think about this too. It's complementary to the list that I just gave you. But if we think about the immune system and its primary job being one of sensing what's going on around us as organisms, whether we're sensing things that are interior and inside or things that are coming from the outside, we might expect that if the tuning of those systems is related to risk for SLE, that we would find variants that are related to signals. And indeed we do when we think of opsonins. There's complement, mannose-binding lectins, C-reactive protein, and perhaps even antibodies that have variants related to SLE. Then there are the sensors we've already talked about, MHC class II. I've alluded to receptors for antibody, the Fc receptors, complement receptors, and TLR. I haven't mentioned MDA5. This is an intracellular receptor sensor for viral products, and it may again be this balance of identifying what might be coming in and challenging the host in sort of a spy versus spy paradigm. In addition to the sensors then, there's integration of those sensors in early signaling and in B cells and T cells. I've listed some of the possibilities for that. And then, of course, the cell response, and this is typically an intermediate response where it might be the interferons and TNF-AIP3. What about predictors and prognosticators? Is all of this work that we're doing in genetics going to take us into that space? Well, I think here we really don't have the information yet to know exactly how this will evolve, but combining GWAS data with functional data types might well elucidate disease mechanisms, as I've alluded to already, and may lead to eventually to a mechanistic rather than a symptomatic patient classification scheme. So finally, what's next? What can we anticipate over the next several years? And I'd like to suggest several things. First of all, with a little bit of perspective. What did we know before this pre-genomic era started? Well, we knew about MHC class II, and we knew about complement components primarily through hereditary deficiencies. What have we learned so far? We've certainly affirmed MHC class II and a variety of components involved in immune complex handling. And we have new things that we've learned, the TLR and the interferon alpha pathway, as I've already alluded to, signaling thresholds, again, as illustrated in the review from Peter. And these signaling thresholds may well involve new inflammatory response intensities. And also we're now learning about viral sensors, MDA5 and RIG1, again with this constant interplay between we as hosts and pathogens that we may be interacting with. And what are some of the new questions? Well, several of the variants raise interesting issues about the process of cell adhesion, or at least cell-to-cell contact, and whether that's sticking, trafficking, or signaling, I think has yet to be worked out. There are a lot of questions about methylation. This is methylation of DNA from an epigenetic perspective, and whether the genetic machinery may alter our quantitative propensity for methylation, and then also what the DNA methylation per se does to the regulatory elements that have genetic variations. And then we must also consider different types of variations, rare variants, CNVs, and some others. Rare variants may often have high penetrance, like the TREX1 variant with an odds ratio that may be as high as about 25. Additional structural variations include copy number variants, insertions, deletions, and some others. Gene regulation is certainly going to be very important, and here the role of epigenetics will be critical. But then we need to pull this all together in terms of system modeling and mechanistically oriented patient classification. I'd like to give you just a couple of quick examples. So different types of structural variations in the genomes. I've mentioned rare variants. We should also not forget splice variants, which may be very important and may impinge on some signaling molecules that are directly involved in the response to immune complexes, and also copy number variations. And I'd like to show you an example that comes from the Fc receptor cluster in humans. For those of you who know the work that I've been involved with for a good number of years, you know that this region is of particular interest to us. And this is a graph which plots in a ratiometric way the genes within this cluster. There is a copy number variant that has been written about quite a bit, and that has to do with CD16B or FCR3B. It's well known. It was first described actually in the 1980s. And if you have a variation along that axis or in that gene, in this particular display that I'm showing here, you will fall on the axis that goes from the lower left to the upper right. I don't know if this actually projects well enough for you to see that there are dots that fall off the diagonal. It really is more than 15% of persons fall off the diagonal, and what that's telling us is that within this cluster, at least 1 out of 6 of us have variations other than those that are classically described and studied. So this is obviously an area for us to do more work. The other possibility I raised was one of modeling, looking at all these variants in a combinatorial fashion, taking a systems approach. I couldn't resist pointing back to a paper that actually I was involved with a few years ago, where we tried to do this with a physical chemist that was at the University of Minnesota at the time. Working from the assumption that if we thought about the constituents of immune complex handling in terms of a systems approach, we might be able to get new insights into the way immune complexes are handled in the quantitative relationships. I'm not sure that we got the new insights. We did get a couple of papers, and at that time that seemed to be the real end game. But where this is taking us is now that we have these extremely large data sets that are becoming available on all of our patients, we have some very new opportunities to look for new relationships amongst the various SNPs and other polymorphisms. Sometimes people call that a data-driven approach, big data, For those of you who follow developments within the NIH, this is now one of 3 major initiatives being supported by the National Institutes of Health. So how do we put all this together? I'd like to suggest a couple of additional questions for us to think about and to work on. First, where is the missing heritability? I think actually that's a false proposition. We know a number of different ways in which we can identify additional contributions to the genetic risk for SLE. But it's important for us to pursue this particular direction. Something I haven't touched upon at all, but we know from our work both in European Americans and African Americans that the genetic architecture of different ancestry groups is different. And understanding that for different backgrounds will be very important for understanding the pathogenesis of SLE. A third question is, if there's lots of overlap across different autoimmune diseases, why are there different autoimmune phenotypes? I think this question is going to be one of the most important, although at the moment exactly how to approach it is not clear. Perhaps one of the ways is to remember that although everything I've talked about has been in the human system, we ought not forget model systems. And we can take the human data, bring those to model systems, and begin to look into what model systems can teach us. Of course, not everything there is absolutely straightforward. Okay. Well, what are some of the significances of the genetic discoveries in SLE? I think, first of all, important insights into pathogenesis. Second, tools and targets to assess differential genetic burden. Third, novel targets for potential therapeutic focus. And finally, we hope there will be markers for disease severity. As we put all those pieces of the puzzle together, I'm sure will enhance our understanding of lupus. And I do understand that one of the themes of this conference is the importance of novel therapeutics and therapeutics applied in a targeted fashion. And I think the genetic variation is going to be a critical part of that puzzle. So with that, I'd like to end thanking many, many people who have been involved in these studies. And indeed, those who are mentioned on this slide are only a few of the many. But in particular, I'd like to acknowledge the Slegion group, the Profile group, which involves multiple centers around the country, but is based at UAB, and the UAB team. Research support has been from the National Institutes of Health, the Alliance for Lupus Research, and many other organizations. And finally, I'd like to thank Howard Hawley. In the introduction, you may have noted that I have a position that bears Howard Hawley's name. He had an autoimmune disease. He's no longer with us. He actually was the founder of the division at UAB. But his commitment to the understanding of the pathogenesis, the terms then he used were terms of biochemistry, not molecular genetics. But his commitment to that understanding is the foundation for moving forward, is one of the things that I hope and I trust drives all of us. With that, I'll thank you very much. And Marta, questions now or at the end? No. Okay, thank you.
We have more or less 4 minutes for questions. If there are any questions from the audience, please. Yehuda, please.
Yeah, that was a very nice introduction. I would like to be a little bit naive and ask you a question, not from a geneticist, but a clinician and maybe a basic researcher. Is it the genetic marker which determines the disease, the autoimmune disease, or is it the genetic marker which determines our reaction to environmental factors, viruses, bacteria, which determines which autoantibody will be produced, and then the disease is determined? I took the liberty recently to publish a paper which is called genetic and autoantibodies. And you didn't mention in your very nice introduction and maps, even one time, maybe once I saw it in your map, that genetic determines autoantibodies, autoantibodies determine the disease.
So I think the answer to your question is both, and it may depend on which marker one is talking about. I hope you all realize we're talking a little bit about philosophy, but one of the reasons I put up the metaphor of the spy versus spy is in that metaphor, one of the spies, you can choose which one you want, is the external environment. And we're always, and we have over time, been adjusting to the external environment. But I think we now have a couple of markers, which I haven't talked about here, but perhaps one that predisposed to end-stage renal disease, where it probably is pretty clear it's the marker per se that's driving the process. So I think what we're going to find is that sometimes the genetic variation is tuning the way our immune system responds to stimuli, and there may be some where, in fact, the determinant itself is driving the process. Dan.
Thanks, that was excellent. And this is sort of analogous to the discussion that you've just had, and that is I'm not entirely convinced that we're dealing with a disease. I mean, the phenotypic variation and the genetic variation reminds me of the situation with dropsy in the 1700s. You know, there's just so many different things contributed to that it took centuries to sort of sort it out. And maybe we're going about it incorrectly by making diagnostic criteria and not just starting from the genetics and working backwards to the clinical phenotype.
Well, the 1700s were just a little bit before my time, so I can't talk specifically about that. But I think, Dan, actually, we both said, perhaps you a little bit more directly than I did, but we're saying very similar things. I think we're now developing the base of information which is going to allow us to define things on the basis of mechanism rather than solely the clinical criteria that we're using. And I think we're going to find very clearly that we're going to segregate into different groups. Whether we call those different diseases, I'm sure there will be many of us involved in committees that will be discussing nomenclature. What this will allow us to do is to use mechanism-based ways of categorizing our patients. And I'm hoping that will also drive more precision-oriented therapeutics as well.
Any more questions? Well, I think we can continue.
Okay, thank you very much.
Bob, thank you very much for an excellent talk.